So today I'm going to tell you about Arabidopsis proteins that are involved both in the regulation of splicing and microRNA biogenesis. These proteins are the nuclear cap binding complex, protein complex, consists of two cap binding proteins, CBP20 and CBP80, and the SERRATE protein, which is a zinc finger containing proteins. I will try to convince you in my talk that these two factors are involved in the communication between the spliceosome and the microRNA biogenesis machinery in plants. In plant, microRNA genes form independent transcriptional units. They are transcribed, all of them, they are transcribed by polymerase II. And as all Pol2 transcript, they obtain the cap structure at the 5' end, and of course the poly-A tail at the 3' end. The characteristic feature of all microRNA transcripts is the presence of a specific stem loop structure in which a microRNA sequence is embedded. In the first step of microRNA biogenesis, the stem loop structures are cut out from long primary transcript called microRNAs, and then in the second step, microRNA star complementary sequence duplexes are cut out from these stem structures. In contrast to animals, in plant, both these microRNA biogenesis steps take place in the nucleus, and both are catalyzed by one single endonuclease dicer-like enzyme DCL1. But to achieve high efficiency and precision in all these cleavages, DCL1 must collaborate with other proteins. And these proteins are CBC, the nuclear cap binding complex, CBP20 and 80, SERRATE, which I introduced before. It's a zinc finger containing protein, and HAL1, which is a double-stranded RNA binding protein. Interestingly, these two guys, CBC and SERRATE, was shown before to be involved in pre-mRNA processing. Of course, CBC is well known because it was described, it was ascribed actually as a splicing factor involved in pre-mRNA splicing. But our group and some other groups described SERRATE as a protein involved in splicing somehow, but the mechanism is still unknown. Okay, so it seems that CBC, the nuclear cap binding complex, and SERRATE, they have double roles in microRNA biogenesis. And you can see here, the best way to see these double roles of SERRATE and CBC is to see, to monitor biogenesis of microRNA. A lot of microRNA primary transcripts are pretty long, and they contain introns. So as you can see here, in the case of microRNA 163 gene, you have a microRNA coding sequence within the first exon, and then you have intron, and then the second intron. Note that this is not protein coding gene, but this is non-coding transcript, okay? So this is just a specific for precursors for microRNA. And if you monitor splicing and microRNA production coming from this kind of transcript, in this case is 163, microRNA 163, you will be able to see that, of course, the production, if you monitor the production of microRNA 163 and splicing of primary transcript of the micro 163 gene in different microRNA biogenesis mutant, HAL1, SERRATE, CBP20, 80, or double mutant CBC, you will realize immediately that the level of microRNA 163 in all this mutant is very low in comparison to the wild type, where I mark here as a 100%. So in all these mutants, the level of mature microRNA 163 is lower. But it's not only this. You can see in the upper panel that when we compare the unspliced and spliced version of microRNA precursors 163, you will see that in SERRATE, CBP20, CBP80, and CBC, that the ratio between spliced and unspliced is changed. So this is the wild-type ratio in the wild-type plants. And then in SERRATE, CBP20 and 80, of course, it's a low level of spliced transcripts. It means that CBC and SERRATE affect the microRNA 163 accumulation and splicing of primary precursors of 163. So, but the question is whether you need the splicing to get microRNA production or not. So it means whether you have any communication, the crosstalk between the microRNA biogenesis machinery and the spliceosome. To check it, first we selected a very useful 163-2 mutant. This is the mutant with disrupted miR163. This is the single gene of this microRNA in the Arabidopsis genome. As you can see here in the selected mutant, there is no production. So this is like knockout mutant, null mutant. There is no production of 163. Fortunately, actually, the phenotype of this mutant is like wild type. You can compare the mutant with the wild type plant. And this is because 163 microRNA is involved in defense responses to pathogen infection. So if you grow your plants without any pathogens, the plants are really happy, and you cannot distinguish between these two phenotypes. Okay, so to check whether splicing is connected with microRNA biogenesis, we prepared two constructs. One, wild type 163 gene with intron, and another one without intron. We introduced both constructs into the 163-2 mutant, remember, without no production of endogenous 163. And we selected two transgenic lines for each construct. As you can see here in the lower panel, the production, this is the wild-type level of 163. This is the level in the mutant. There's no production of 163, no accumulation. But then, when we introduce the wild-type gene, the level is almost the same like the wild-type. But when we introduce the intron-less gene, the level dropped down dramatically. So you can observe just 30-something%, more than 30%, but less than 40% of wild-type production of 163. And of course, we checked the level of precursors and check splicing isoforms. What's important is that if transcript is produced from intron-less gene, we observed accumulation of precursors. So it's something wrong with the microRNA biogenesis if the transcript does not contain introns. So you can see the accumulation of this. So from this experiment, it's clear that introns are important somehow for production of microRNA from intron-containing genes. But the question is whether we need just intron sequence or maybe splicing. So to answer this question, we prepared some additional constructs, and in this construct, we mutated the 5' splice site in the microRNA 163 gene, 3' splice site of both splice sites together, and again we introduce all these constructs into the 163-2 mutant without endogenous production of 163. Again, for each construct, we selected two independent transgenic lines, and we tested the level of 163. As you can see here in this lower panel, when we mutated the 5' splice site, the level dropped down to 30-something%. But similar mutation, but at the 3' splice site, the level of microRNA accumulation was almost the same like in the wild type, a little bit lower, but still comparable. And in the case of constructs, if mutated both splice sites, the level is even lower. It's something below 20%. And as you can see here, when we have mutated both splice sites, the transcript is not stable. And we know that this is not because of transcription. This is just because instability of the transcript. In the case of 5' splice site mutant and 3' splice site mutant, in both cases you observe accumulation of transcripts, this unspliced transcripts. So it's clear from this experiment that not splicing per se, but somehow the 5' splice site is important for this interaction between the microRNA biogenesis machinery. And what's even more interesting is that in the case of microRNA 163 gene, you have two polyadenylation sites. One is which we called proximal one within intron, and the second is the distal one here. So you have two polyadenylation sites. In the wild-type plants, you use 43%. In 43% of cases, the proximal site is used, and then the rest is the distal one. But when we mutated the 5' splice site in this construct, we observed actually increased level of use of this proximal splice site. And the same was true in the case of mutated both splice site 5' and 3'. We didn't observe any changes when we mutated the 3' splice site. Thus, the 5', the active 5' splice site influences the choice of polyadenylation sites. So you need active 5' splice site to block somehow the proximal polyadenylation site. Okay, so the question was whether this is relevant. It means whether it has any biological sense, this correlation or crosstalk between the microRNA biogenesis and biogenesis machinery and the spliceosome. So first, we identified one of a target of 163. I do not have time to explain exactly how we did this, but a target S-adenosyl L-methionine-dependent methylotransferase turned out to be a real target of 163. And as you can see here, the level of the target increased. I'm sorry. The level of the target increased in all this microRNA biogenesis mutant we tested before. Why? Because in all these mutants tested before, the level of 163 is lower. So this is why the level of target in all these mutants is higher. This is a proof that we are looking at a real microRNA 163 target. Okay. So I told you at the beginning that microRNA 163 is involved in defense responses to pathogen infection in Arabidopsis. So we decided to check whether infection with Pseudomonas syringae of two lines, wild type and so the line expressing 163 from the wild type gene and expressing from the gene with mutated 5' splice site, whether the response will be exactly the same. And as you can see here, the response was completely different. In the case of wild type, the plants expressing 163 from the wild type gene, you observed the accumulation after 24 hours of infection, after infection of 72, accumulation of 163 microRNA. And of course, we observed the lower level during this, upon infection, during infection time, the level of precursors. Why? Because the precursors was processed immediately into the mature microRNA. But in the case of mutant, the mutant plants means that they have 163 gene, but with mutated 5' and 3' splice site. And as you can see here, there is no response at all, so the level is still the same, 30-something% in all cases. And the primary transcript level increased even. So the response, the plant response is completely different. And you can check it even at the level, check the level of target mRNA, and you see here that the target decreased upon infection because, of course, we observed the higher level of 163, but in the case of mutated version, the target even increased. So it's completely, the response to pathogen infection is completely changed. Okay, so what we think about the mechanism of correlation, or about the crosstalk between the microRNA biogenesis machinery and the spliceosome. We think that at the beginning of recognition of primary transcripts for microRNA transcripts containing introns, you have a complex of CBP80, 20, and SERRATE, and this complex helps somehow U1 to recognize the 5' splice site. Of course, you have SR proteins that help or stabilize actually U1 binding to the 5' splice site. Actually, I talked about it last year, about this SR protein stabilizing U1 binding. And of course, you have some influence of the 3' splice site. So then you have this kind of complex. Maybe I should mention here that the presence of U1 at the 5' splice site block the proximal polyadenylation site in this case. So the distal polyadenylation site is used. Then after these interactions, you have stabilization of the stem loop structure, and then the stem loop structure can be recognized by DCL1 and HIL1, and SERRATE can probably, or CBP2080, can recognize the complex. It's not anymore needed for U1 stabilization. What we know about all this interaction? We know that CBP20 and CBP80 interact with SERRATE. And as you can see here in this bimolecular fluorescent complementation assay in Arabidopsis protoplasts, CBP820 interacts with SERRATE. This is the nucleus. This is the nucleus, and these bright spots are just the dicing bodies. This is the structures in which microRNA processing is taking place. Okay, and the same is true for CBP80 and SERRATE. So both proteins of CBC, the CBC complex, interact with SERRATE. We proved this using a pull-down assay with recombinant protein, and again, I do not have time to go into details, but you can see that SERRATE interact in this in vitro assay with CBP20, with CBP80, and SERRATE interacts with the whole complex as well. So that's clear that SERRATE can interact with both subunits of CBC. Moreover, we actually wanted to see the interaction from U1 SNRP. So we tested actually all possible possible U1 SNRP proteins to interact with SERRATE. Strong interactors and weak interactors, whatever, and connected with U1 SNRP proteins. And what's clear, this is in the EASE2 hybrid system, and what's clear from this assay is that PRP39, PRP40, and interact strongly with SERRATE. And in the case of LAC7, only one variant of LAC7 in Arabidopsis, LAC7RL, interacts with much, I mean, the interaction is much weaker. Okay, so we have some U1 interactors to prove our hypothesis and the model of interaction between the spliceosome and the microRNA biogenesis machinery. Of course, almost all of these interactions we prove by pull down with recombinant proteins. So we are pretty sure that I'm not saying that they are functional, all these interactions. This is something we have to prove, but at least they are possible somehow. Okay, and that's it. Conclusions. Introns in prime microRNA stimulate production of mature microRNA. Induction of microRNA 163 upon pathogen infection depends on functional splice sites of the gene. This is what probably we proved. U1 SNRP plays a role in the selection of polyadenylation site. And what we think is that the crosstalk between CBC, SERRATE, and U1 SNRP plays a crucial role in regulation of microRNA splicing. And I didn't have time to show you data that exactly the same interaction is in the regulation of pre-mRNA splicing. So we believe that the CBC, SERRATE, and U1 interaction is important for the selection of the 5' splice site in alternative splicing, the 5' splice site of the first intron. This work was performed in the Institute of Molecular Biology and Biotechnology of the Adam Mickiewicz University in Poznan in Poland. And Zofia Szwejkowska-Kulińska was a co-PI of this study. And actually, Dawid Bielewicz did most of the experiments I presented here. And this is our collaborators, Andrea Barta and Maria Kalina from University of Wien, and Frank Vasquez and David Windels from Basel. Thank you very much.